Hey everyone, welcome back to my channel. In the first part of respiration and circulation chapter, we covered the respiration part that is available on my channel, and now we will start from circulation part. Circulation in animals. In higher animals, the circulation is carried out by special fluids, blood and lymph. Blood vascular system. Higher animals from Annelida to Chordata have a special circulating fluid, the blood which is pumped to the tissues by the heart through the blood vessels. Type of blood vascular system, open circulation. In animals having an open circulation, blood is circulated through the body cavities, that is hemocoels. The visceral organ lie in the blood-filled body cavity. Exchange of material takes place directly between blood and cells or tissue of the body. The blood flows with low pressure and usually does not contain any respiratory pigments like hemoglobin, so it does not transport any respiratory gases. Example, arthropods that you have studied in 11th standard, cockroach and mollusk. This is the diagram that you have to observe. You have to label it. Then we have closed circulation. In all the vertebrates, higher mollusks and annelids, blood is circulated over the body through a network of blood vessels. In this type of circulation, blood flows within the blood vessels and does not come in direct contact with cells and body tissues. Exchange of material between blood and body tissue is through an intermediate fluid called lymph. Blood vessel with high pressure and contains respiratory pigments like blood flows with high pressure and contains respiratory pigments like hemoglobin for transportation of respiratory gases. The closed circulation can be divided into two main types, that is single and double circulation. So let's see single circulation. In single circulation, the blood passes through heart only once during each cycle, as in fishes. Deoxygenated blood is pumped from heart towards gills where it undergoes oxygenation. This oxygenated blood moves towards various body parts, gets deoxygenated again and returned back to heart for next cycle. This is the single circulation diagram given. You can observe it. Since the heart of fish carries only deoxygenated blood, it is also called venous heart. And then we come to double circulation. In double circulation, blood passes through heart twice during each cycle. It occurs in birds and mammals. In these animals, the heart pumps deoxygenated blood to lungs for oxygenation and returns to heart as oxygenated blood. This is pulmonary circulation. Deoxygenated blood is pumped from heart towards various body parts except the lung and returned back to heart as deoxygenated blood. This is systematic circulation. Human heart shows double circulation. So this is the diagram of double circulation. You can observe it. Circulatory system in human. The human circulatory system is composed of blood vascular and lymphatic system. Blood vascular system. In human being, it consists of blood, heart and blood vessels. It is responsible for various functions like transport, homeostasis and protection. Blood composition and coagulation. Study of blood is called hematology. An average adult has about 4 to 6 liters of blood. It is a red colored fluid connective tissue derived from embryonic mesoderm. It is slightly alkaline, pH is 7.4, salty and viscous fluid. It is heavier than water. It has two main components, the fluid plasma, that is 55%, and the formed elements, that is blood vessels, blood cells, 44%. These can be separated by centrifugation. Plasma. It constitutes 55% of the blood. It is a straw colored, slightly. So we have water, that is 90%, protein 7 to 8%, inorganic salt 1%, other things like food, waste, regulators, anticoagulants, cholesterol and antibodies, dissolved gases is 1 to 2%. Then we have formed element. The blood cells that are produced in the body are collectively called formed elements. Human blood contains three types of formed elements as red blood corpuscles, RBCs, white blood corpuscles, WBCs and platelets. Then we come to red blood corpuscles, that is erythrocytes. Erythrocytes are the most abundant cells in the human body. They are circular, biconcave and enucleated. In camel and llama, they are nucleated. The red color or RBCs is due to an oxygen carrying pigment, the hemoglobin, in their cytoplasm. In males, their average number is about 5.1 to 5.8 million per microliter, and in females about 4.3 to 5.2 million microliter. This is called total RBC count. The average lifespan of RBC is 120 days. The process of formation of RBCs is called erythropoiesis. RBCs are produced from hemocytoblasts or reticulocytes. The erythropoietic organ of the fetus is the liver and spleen, and in the adult it is mainly the red bone marrow. Vitamin B12, folic acid and heme protein are required for production of RBCs. The old and worn out RBCs are destroyed in the liver and spleen. Spleen is also called the graveyard of RBCs. Condition with increase in the number of RBC is called polycythemia, and with decrease in number of RBC it is called as erythrocytopenia. The hormone erythropoietin produced by the kidney cells stimulates the bone marrow for production of RBCs. Mature erythrocyte is devoid of nucleus, mitochondria or other membrane bound cell organelles. Its cytoplasm, that is stroma, is rich in hemoglobin and O2 carrying proteinaceous pigment that gives red color to the RBCs and blood. It also contains an enzyme carbonic anhydrase. Erythrocytes are responsible for the transport of respiratory gases, O2 and CO2, maintaining pH and viscosity of blood. They also contribute in the process of blood clotting. The hematocrit is ratio of the volume of RBCs to total blood vessels, total blood volume of blood. It is different for men and women. Okay, each erythrocyte approximately contains 270 million molecules of hemoglobin. Normal content of hemoglobin in blood of men is about 14 to 12 gram, and in women it is about 13 to 15 gram. Condition with less number of RBCs or less amount of hemoglobin or both is called anemia. Each molecule of hemoglobin is a protein iron complex. It consists of 4 polypeptide globin chain, alpha and 2 beta chains. An iron porphyrin, that is heme group, is attached to each chain and all 4 chains are bound together. Each heme group carry out one O2 molecule, and thus one hemoglobin molecule can carry 4 O2 forming oxyhemoglobin. CO2 interacts with amino acids residues of globin chain and forms carboamino hemoglobin. After hemolysis, hemoglobin is broken down. Its globulin part is broken to recycle the amino acids. Iron of heme group is stored as ferritin in the liver, and porphyrin group of heme is converted into green pigment biliverdin and then into red orange colored bilirubin. These pigments, mainly bilirubin, are added to bile and finally removed out of body along with feces. Then we have white blood corpuscles, that is leukocytes. Leukocytes are colorless, nucleated and amoeboid cell larger than RBCs. Due to their amoeboid movement, they can move out of the capillary walls by a process called diapedesis. A normal adult has an average 5,000 to 11,000 WBCs per millimeter cube of blood. Okay, decrease in number of WBCs, that is less than 4,000, is called leukopenia, common in HIV, AIDS and TB patients or those exposed to radiations, shocks, etc. Temporary increase in the number of WBC is called as leukocytosis. It is due to infection. It also occurs during pregnancy and in newborn babies. Uncontrolled increase in number of WBCs is a type of blood cancer called leukemia. WBCs are mainly concerned with defense mechanisms, that is protection. Types of WBCs. These are colorless, irregular nucleated cells and show polymorphism, exist in variable forms. They can be classified in two main types, such as granulocytes and agranulocytes. Granulocytes. Okay, so these are WBCs with a granular cytoplasm, also called polymorphonuclear leukocytes, PMN cells. They have lobulated nuclei in different shapes. Granulocytes are formed from myeloid stem cells. Myeloid stem cells, and once formed, do not divide. Granulocytes constitute about 72% of total WBCs. Granules are actually secretory vesicles which contain various secretions, enzymes, etc. Depending upon staining property of the granules, these granulocytes are classified into three types as neutrophils, basophils, and acidophils. Let's see the diagram. So this is neutrophil, then B is basophil, C is eosinophil, we have lymphocyte and monocyte. These diagrams are very much important. The questions can come directly from the diagram also. So let's see neutrophils. Granules are very fine, large in number, evenly distributed and stained with neutral stains, dyes. Neutrophils are about 70% of total WBCs. These cells are spherical and nucleus is several lobed, that is 2 to 7. These are able to perform amoeboid movements and phagocytosis. They are responsible for destroying pathogens by the process of phagocytosis. Pus is mixture of dead neutrophils, damaged tissues and dead microbes. Let's come to basophils. Basophils are also called as cyanophils. These cells have very few granules of large size and stain with basic stains like methylene blue. Basophils are non-phagocytic, small spherical cells and are about 0.5 to 1% of total WBCs. Nucleus is twisted. They are present in infected and allergic conditions only. Basophils secrete heparin, histamine and serotonin. Then we come to eosinophils or acidophils. Acidophils contain lysosomal granules that are stained to red color with acidic stains like eusin. Eosinophils are about 1 to 3% of total WBCs. Nucleus is bilobed. They destroy antigen-antibody complex by phagocytosis. Their number increases in allergic condition and they show antihistaminic property. They are also responsible for detoxification as they produce antitoxins. B is agranulocytes. Agranulocytes are about 28% of total WBCs. Cytoplasm of these leukocytes is without granules. They are formed from lymphoid stem cells and can divide by mitosis. Nuclei of agranulocytes are large in size but are not lobulated like the granulocytes. There are two types of agranulocytes, lymphocytes and monocytes. Lymphocytes are the smallest of all WBCs and have a large spherical nucleus. They constitute about 25 to 30% of total WBCs. Depending upon function, two types of lymphocytes are present as B lymphocytes and T lymphocytes. B lymphocytes mature in bone marrow and are responsible for antibody production or humoral immunity. It is a highly specific antigen. Antibody immunity T lymphocytes mature in thymus and are responsible for cell-mediated immunity. Helper T cells, killer T cells, memory T cells and suppressor T cells are four main subtypes of T lymphocytes. Then we come to monocytes. Monocytes are the largest of all the WBCs. Its nucleus is large in bean or kidney shape. They form 3 to 5% of WBCs, that is white blood cells. Monocytes are actively motile and give rise to macrophages. They are mainly phagocytic and destroy the bacteria and dead or damaged tissue by phagocytosis. Then we come to thrombocytes or platelets. Thrombocytes are cellular fragments formed from the large cell called megakaryocytes. These are produced in bone marrow. They are very small, oval-shaped cells, fragments without nucleus. Normal count of thrombocytes in human blood is about 2.5 to 4.5 lakh per millimeter cube of blood. If number of thrombocytes decrease than normal condition, it is called as thrombocytopenia. This condition causes internal bleeding. Platelets secrete platelet factors which are essential in blood clotting. They also seal the ruptured blood vessels by formation of platelet plug or thrombus. They secrete serotonin, a local vasoconstrictors. This is the blood schema diagram. Then we come to blood clotting or coagulation of blood. Clotting or coagulation is the process of converting the liquid blood into a solid form. This process may be initiated by contact of blood with any foreign surface, that is intrinsic process, or with damaged tissue, extrinsic process. Intrinsic and extrinsic processes involve interaction of various substances called clotting factor by a stepwise or cascade mechanism. There are in all 12 clotting factors numbered as 1 to 13 factor. 1 to 13. Factor 6 is not in active use. Interaction of these animals in a cascade manner lead to formation of the enzyme thrombin. Thromboplastin helps in the formation of enzyme prothrombinase. This enzyme inactivates heparin and it also converts inactive prothrombin into its active thrombin. Thrombin converts soluble blood protein fibrinogen into insoluble fibrin. Fibrin forms a mesh in which platelets and other blood cells are trapped to form the clot. Blood clotting occurs as shown in the diagram. Let's come to heart. Heart is the main pumping organ of the circulatory system. It is reddish brown in color, hollow muscular organ roughly the size of one's fist. Its average weight is about 300 grams in males and 250 grams in females. It is conical in shape and lies in mediastinum, that is the space between two lungs. It is broader at upper end and conical at lower end. Conical end is slightly tilted to left side and rest above the diaphragm. Heart is enclosed in a membranous sac called pericardium. Pericardium is formed of two main layers, outer fibrous and inner serous pericardium. Serous pericardium is soft, moist and elastic. It is formed of squamous epithelium and is further divisible into two layers as parietal and visceral layer. Parietal and visceral layer of serous pericardium are separated by a pericardial space. This space is called filled with pericardial fluid. It is about 50 ml, which acts as a shock absorber and protects the heart from mechanical injuries. It also keeps the heart moist and acts as a lubricant. Heart wall. The heart is mesodermal in origin. Its wall is formed of three layers: outer epicardium, middle myocardium, and inner endocardium. Epicardium is thin and formed of a single layer of flat squamous epithelium resting on basement membrane. Myocardium is the middle thick layer formed of cardiac muscles. Endocardium is a single thin layer formed of squamous epithelium. The epicardium and endocardium are protective in function, whereas myocardium is responsible for contraction and relaxation of heart. External structure of heart. The human heart is four-chambered. The two superior chambers are called atria or auricles and inferior two are called ventricles. Externally, the atria are separated from ventricles by a transverse groove called coronary sulcus or atrioventricular groove. The two ventricles are externally separated from each other by two grooves, the anterior and posterior interventricular sulci. Coronary arteries and coronary veins run through these sulci. Pulmonary trunk arising from the right ventricle and aorta from left ventricle are present on anterior surface of the heart. The diagram is given. You can see external and external structure, posterior and anterior. The pulmonary trunk bifurcates into right and left pulmonary arteries. Aorta is divisible into three regions as ascending aorta, systematic aortic arc, and descending aorta. The ligamentous arteriosum joins pulmonary trunk and aortic arc. It is the remnant of an embryonic duct called ductus arteriosus. The aortic arc gives out three arteries, namely brachiocephalic, innominate artery, left common carotid, and left subclavian. The right atrium receives superior and inferior vena cava along its dorsal surface. Pulmonary vein opens into left atrium along the dorsal surface of heart. Now let's come to internal structure of heart. Atria. These are thin-walled receiving chamber of heart. They are separated from each other by interauricular septum. Interauricular septum has an oval depression called fossa ovalis. It is a remnant of the embryonic aperture called foramen ovalis. Superior vena cava or precaval, inferior vena cava, postcaval, and coronary sinus open into the right atrium. Opening of the postcaval is guarded by a eustachian valve, while the Thebesian valve guards the opening of coronary sinus into right atrium. Four pulmonary veins open the left atrium into the left atrium. These openings are without valves. Both the atria open into the ventricles of these respective sides by atrioventricular apertures. These openings are guarded by cuspid valves. The tricuspid valve is present in the AV aperture, and bicuspid valve, also called as mitral valve, is present in the left AV aperture. All these heart valves help in maintaining a unidirectional flow of blood. They also avoid backflow of blood. Now come to ventricles. These are inferior, thick-walled pumping chambers of the heart. The right and left ventricles are separated by an inner ventricular septum. Wall of the left ventricle is more muscular and about 3 times thicker than the right ventricle. Inner surface of the ventricles show several ridges called columnae carni or trabeculi carni, which divide the lumen of ventricles into small pockets of fissures. The lumen of ventricles also shows inelastic fibers called chordae tendineae. These attach the bicuspid and tricuspid valves to the ventricle wall and regulate their opening and closing. The right ventricle opens into the pulmonary aorta and left ventricle opens into the aorta. These openings are guarded by 3 semilunar valves each. These valves present the backward flow of blood into the ventricles. Pumping action of heart. The heart acts as the main pumping organ of the circulatory system. The pumping action is brought about by a rhythmic contraction and relaxation of the cardiac muscles or heart muscles. Contraction of heart muscles is systole and relaxation of heart muscles is diastole. A single systole followed by a diastole makes one heartbeat. The heart beats 70 to 72 times per minute. This is called heart rate. During each heartbeat, ventricles pump about 70 ml of blood. This is called stroke volume. It means heart pumps about 72 into 70 ml, which is approximately 5040 ml, approximately 5 liters of blood per minute. This is called cardiac output. Conducting tissue of heart. The human heart is myogenic, that is, the heart is capable of generating a cardiac contraction independent of nervous input. It also shows out rhythmicity, that is, it can generate its own rhythm by specialized muscles. A specialized cardiac musculature called the nodal tissue is distributed in the heart. A part of this nodal tissue is present in the upper right corner of the right atrium. It is called SA node or sinoatrial node. It lies at the base of opening of superior vena cava. Another mass of nodal tissue, the modified muscular fibers, also called autorhythmic fibers, control the beating rate of heart. Conducting tissue consists of SA node, AV node, bundle of His, and Purkinje fibers. Conducting system of the heart. SA node, sinoatrial node, is present in the right atrium. It acts as a pacemaker of heart because it has the power of generating a new wave of contraction and making the pace of contraction. SA node passes the contraction to the left ventricle and also to the AV node. AV node, that is atrioventricular node, is present in the right atrial wall near the base of interatrial septum. It acts as a pace setter of heart. Bundle of His or Tavara branches start from AV node and pass through interventricular septum. Bundle of His forms 2 branches, the right and left bundles, one for each ventricle. These branches form network in ventricular walls and these are called Purkinje fibers. Bundle of His and Purkinje fibers separate impulses in ventricles. As a result, both the ventricles contract simultaneously. Working mechanism of human heart. Cardiac cycle. Human heart alternately contracts and relaxes. Contraction is called systole and relaxation is called diastole. Atria and ventricle contract alternately. Consecutive systole and diastole constitute a single heartbeat or cardiac cycle. It is completed in 0.8 seconds. On an average, 72 beats are completed in 1 minute in an adult at rest. Atrial systole. Right atrium receives deoxygenated blood and left atrium receives oxygenated blood. When both the atria are completely filled with blood, pressure is exerted on the wall. In response to this pressure, SA node gets excited and generates cardiac impulse. Due to these cardiac impulse, muscles in the atrial wall contract, causing atrial systole. During atrial systole, blood is pumped into ventricles. Blood is prevented from going back to the veins and coronary sinus by eustachian and Sebacean valve respectively. After completing systole, the atria go into diastole. In normal condition, atrial systole is for 0.1 second and atrial diastole is for 0.7 second. Ventricular systole. The impulse which started from SA node now reaches the AV node and it gets excited. AV node sends impulse to bundle of His, from bundle of His to Purkinje fibers. Purkinje fibers spread impulses all over the wall of ventricles. Due to this, ventricular wall contracts, causing ventricular systole. During ventricular systole, right ventricles pump deoxygenated blood into pulmonary trunk and left ventricles pump oxygenated blood into aorta. During ventricular systole, the cuspid valves close both the atrioventricular apertures, preventing blood flow into atria. Lub sound is heard. In normal condition, ventricular systole lasts for 0.3 second and ventricular diastole lasts for 0.5 second. During ventricular diastole, semilunar valves are closed, preventing backflow of blood from pulmonary trunk and systematic aorta into ventricles. Dub sound is heard. For about 0.4 second, both atria and ventricle are in diastole. When all the chambers of heart are in diastole, this condition is called joint diastole or complete diastole. Thus, duration of one cardiac cycle is 0.8 second. The diagrammatic representation of cardiac cycle is given. Atrial systole is 0.1 second, ventricular systole is about 0.3 second, and then joint diastole is about 0.7. Okay, I'm wrong. 0.4 seconds. So joint diastole. Now let's go to blood vessels. There are 3 main types of blood vessels in the human circulatory system, namely arteries, veins, and capillaries. Arteries. These blood vessels carry blood from various parts of organ of the body. There they branch into arterioles and further into fine capillaries. They normally carry oxygenated blood to all part of the body except pulmonary artery, which carries deoxygenated blood. They are usually situated deep in the blood. They are usually situated deep in the body except a few like the radial, brachial, and femoral, which are superficially located in a TS of artery. Its walls show 3 layers: tunica externa, tunica media and tunica interna. The outermost tunica externa is a thick, tough layer of collagen fibers. The tunica media is made up of smooth muscles and elastic fibers. This thick muscular and elastic layer makes the arterial wall pulsatile. The innermost tunica interna is a single layer of flat, compact endothelial cells surrounding the lumen. The angular margin around the lumen show tessellations. Arterial lumen is devoid of valves and blood flow through it rapidly with high pressure. Veins are thin-walled, mostly superficial vessels which carry blood from the organ towards the heart. The capillaries around the various organ joints to form the veins, except for the pulmonary vein or other vein of the body, carry deoxygenated blood towards the heart. Portal vein. A portal vein, example hepatic portal vein, differs from the other normal veins in that it starts as a capillary from one organ and capillarizes in some intermediate organ, example liver, before taking the blood towards the heart. Histologically, the veins also show the three layers like in the arteries, the tunica externa, tunica media and tunica interna. However, the tunica media is comparatively thinner and their lumen is wide and narrow. Internal valves at regular intervals can be seen. Blood flows with flow pressure and valves prevent backflow of blood. Capillary. These are a network of minute blood vessels. They are thin-walled, having a single layer of flat squamous epithelium resting on a single basement membrane. They are mainly involved in exchange of materials. Wall of capillaries is formed of single layer of squamous epithelium and it is stretchable. Blood flow through the capillary under high pressure. Wall of capillaries bear small endothelial pores of or fenestrae through which blood vessels can escape by the process called as diapetesis. Come to pulse. It is a series of pressure waves that travel through the arteries during ventricular systole. It is the strongest in arteries closer to the heart and gradually becomes weak in artery away from the heart. It can be felt easily in the superficial arteries like radial artery in the wrist and carotid artery in the neck. The pulse can be felt at particular points on the body. All locations where the pulse can be felt are shown in the figure. So this is the figure. Facial, temporal, carotid, branchial, radial, femoral, dorsalis pedis, popliteal, posterior tibial. Pulse rate is equal to heart rate. Pulse rate higher than normal above above 100 beats per minute is called tachycardia, and slower pulse rate below 60 beats per minute than normal is called bradycardia. Come to blood pressure. The pressure exerted by blood on the wall of the blood vessel is called blood pressure. It is measured by sphygmomanometer. Okay, sphygmomanometer. Okay. Let's start blood pressure. The pressure exerted by blood on the walls of the blood vessel is called blood pressure. It is measured by sphygmomanometer. It is usually measured from the arteries. Arterial blood pressure. Pressure exerted by blood on the walls of artery is arterial blood pressure. Pressure on arterial wall during ventricular contraction is systolic pressure. For a normal healthy adult, the average value is 120 mm of Hg. Pressure on arterial wall during relaxation of ventricle is diastolic pressure. For a normal healthy adult, it is 80 mm of Hg. BP is equal to SP by dP, which is 120 by 80 mmHg. Blood pressure is normally written as 120 by 80 mm of Hg. Difference between systolic and diastolic pressure is called pulse pressure. Normally it is 40 mm of Hg. Deviations from normal blood pressure value indicates malfunctioning of the heart. It may be due to high or low blood volume, arterial inelasticity or hardening of arteries, deposition of fat like cholesterol in the arteries, that is atherosclerosis, renal disease and emotion induced hormonal change, obesity, etc. Blood pressure lower than normal, that is below 90 by 60 mm of Hg, is called hypotension, and blood pressure higher than normal, that is above 140 by 90 mm Hg, is hypertension. Various factors that affect the blood pressure are cardiac output, peripheral resistance, blood volume, length and diameter of blood vessels, viscosity of blood, age, gender, venous return, sleep, emotion, exercise, anxiety, etc. Normal cardiac output is 5 liter per minute. Increase in cardiac output increases systolic pressure. Peripheral resistance depends upon the diameter of blood vessels. Decrease in diameter of arterioles and capillaries under the effect of vasoconstrictors like vasopressin or ADH cause increase in peripheral resistance and thereby increase in blood pressure. Blood loss in accidents decrease blood volume and thus the blood pressure. Blood pressure is directly proportional to viscosity of blood. Blood pressure increases with age due to increase in inelasticity of blood vessels. Amount of blood brought to the heart via the veins per unit time is called venous return, and it is directly proportional to blood pressure. Blood pressure is also directly proportional to the total length of the blood vessels. Blood pressure can also be affected by vasoconstrictions or vasodilations. Females have slightly lower blood pressure than males her age before menopause. However, the risk of BP increases in the females after menopause sets in. Measurement of blood pressure. Blood pressure is measured with the help of an instrument called sphygmomanometer. This instrument consists of inflatable rubber bag cuff covered by a cotton cloth. It is connected with the help of tubes to a mercury manometer on one side and a rubber bulb on the other side. During measurement, the person is asked to lie in a sleeping position. The instrument is placed at the level of heart and the cuff is tightly wrapped around the upper arm. The cuff is inflated till the brachial artery and is blocked due to external pressure. Then pressure in the cuff is slowly lowered till the first pulsatile sound is heard. At this moment, pressure indicated in manometer is systolic pressure. Sounds heard during measurement of blood pressure are called as Korotkov sounds. Pressure in the cuff is further lowered till any pulsatile sound cannot be heard due to smooth blood flow. At this moment, pressure indicated in manometer is diastolic pressure. An optimal blood pressure level reads 120 by 80 mm of Hg. Hypertension. Persistently raised blood pressure higher than the normal is called hypertension. 140 by 90 mm Hg is called as a threshold of hypertension. And the 180 by 20 mm Hg and higher readings are dangerous to the health. It may damage the heart and brain and kidney. Under the condition of hypertension, heart uses more energy for pumping, which causes angina pectoris, the chest pain due to lowered blood supply to cardiac muscles, and may lead to myocardial infarction. Yes, there are more chances of brain hemorrhage due to hypertension, as arteries in brains are less protected by surrounding tissues as compared to other organs. In kidney, hypertension may cause kidney failure. Coronary artery disease, it is also known as atherosclerosis. In this, calcium, fat, cholesterol and fibrous tissue gets deposited in blood vessels and supplying blood to the heart muscles, making the lumen narrow. Now we have angina pectoris. It is the pain in the chest resulting from a reduction in the blood supply to the cardiac muscles because of atherosclerosis or arteriosclerosis. It is characterized by severe pain and heaviness in the chest. The pain may spread to the neck, lower jaw, left arm and left shoulder. The pain usually results from exertion, where there is more demand of oxygen by the heart but supply does not meet the requirement. Angiography. X-ray imaging of the cardiac blood vessels to locate the position of blockages is called angiography. Depending upon the degree of blockage, remedial procedures like angioplasty or bypass surgery are performed. In angioplasty, a stent is inserted at the site of blockage to restore the blood supply, while in bypass surgery, the atherosclerotic region is bypassed with part of vein or artery taken from any other suitable part of the body like hands or legs. Heart transplant and silent heart attack is deleted from your portion, so I'll directly move on. Electrocardiogram. Graphical recording of electrical variations detected at the surface of body during their propagation through the wall of heart is called electrocardiogram, ECG. This recording may be in the form of printout or on-screen display. The instrument used for this recording is the ECG machine or electrocardiograph. This instrument detects and amplifies the signals. Various electrodes are used for recording of signals. 4 electrodes are positioned on limbs, 2 on arms and 2 on legs. These are limb electrodes. 6 electrodes are positioned on chest. These are chest electrodes. In a normal recording, 3 different waves are recognized as P wave, QRS complex and T wave. P wave is a small upward deflection from baseline of graph. It represents the atrial depolarization. Very important, P wave represents atrial depolarization. The QRS complex starts as a slightly downward reflection from baseline, continues a sharp and large upright wave and ends as a downward wave. QRS complex represents the ventricular depolarization. Again, QRS complex represents the ventricular depolarization. Then we have T wave. T wave is a small, wide and upwardly elevated wave. It represents ventricular repolarization. ECG helps to diagnose the abnormality in conducting pathway, enlargement of heart chambers, damages to cardiac muscles, reducing blood supply to cardiac muscles, and causes of chest pain. A physician can find out the defect in the heart by examining the wave pattern and the time interval between them. Lymphatic system. Lymphatic system consists of lymphatic vessels, some organs and tissues. The word lymph means clear water and is a fluid connective tissue with almost similar composition to the blood except RBCs, platelets and some proteins. Fluid from intercellular space of the body tissue enters into the lymphatic vessel. From here it is discharged into the blood vessels through the thoracic duct and the right lymphatic duct. This is the lymphatic system given. Okay, so the chapter ends here, I think. Yes. Thank you for watching the video. If you liked it, please do like, share and subscribe to my channel. And please tell me which chapter's audiobook you want in the next video. Thank you. Bye-bye.